Okay, good morning, everybody. My name is Dave Muir. I'm going to be teaching you this semester. So welcome to the fall semester 2025, St. Francis College. This is Bio 3300, Microbiology. If any of you want to be studying accounting or anything else, this is not the class for you. So this is a bit unusual because we will usually cover topics with me starting off the topic face-to-face in class and then continuing with recording to conclude topics. But today, because our semester starts on a Wednesday and your class is on a Monday, I'm just going to record this quick class on introduction and history. And then I'll meet with you again, of course, on Monday. And on Monday, we'll go over the class details, rules, regulations, etc., etc. There's only one thing I have to say about that, and that is please, would you make sure that you read the 3 short papers which I have posted to Canvas. Please read those carefully because it will explain to you some of the rules that I have in my class, which are enforced quite strictly. I know that these are cause some consternation, but if you read those 3 short papers and listen to the information that I give you in addition, you will understand why I have those rules in place. They are there for a reason. And it is to improve your learning experience. We'll talk more about that on Monday. Okay, so today I am just going to cover very briefly the development of the science of microbiology. And I want to point out that this is for a number of reasons. It's not only important that you hear some of the important figures and their important findings in the history of microbiology, but that you understand that that microbiology is a science. It developed actually from purely observational studies. People did very often as a hobby, just looking at things and trying to explain how they occurred. But the science developed with people who developed scientific method for examining the study. They proceeded by hypothesizing, experimenting, and then proving aspects of microbiology so that it developed as a true science, not just as observation. Okay, so we'll first of all, let me just state that we need to have a definition of what a microbe is. And a microbe is anything which is less than 0.1 millimeters. And the reason we set that definition 0.1 millimeters is below 0.1 millimeters, our eyes are unable to discern the boundaries of an object. In other words, anything below 0.1 millimeters, our eyes are simply not constructed to resolve it so that we can discern one side from the other. We cannot tell its shape. We cannot tell its dimensions, nothing. At the very most, all that we can do is observe it as a point under certain circumstances. And most things below 0.1, we cannot see at all, even as a point. They are invisible to us because our eyes can't resolve them. So if we want to see them, then we have to use a special apparatus in order to do so. And that apparatus, the fundamental apparatus, is a lens. A lens is something which is able to magnify. In fact, what it is doing is it's enabling us to see below 0.1 millimeters by expanding the apparent size of the object. Initially, well, lenses have been known for a very long time. The Romans actually used lenses, but the glass that they made them from was relatively crude, and lens making actually only became highly refined in the 16th and 17th centuries. And at that time, people very often just did it as a hobby. They made lenses and looked at things. And they almost entirely depended upon single lenses. So that distinction will become apparent to you, because when you do some reading that I have set for you, which can wait until next week, I have set for you a reading of the sections in your text about microscopy. And microscopy nowadays largely depends on using compound lenses, many lenses put together to increase the magnifying power. The initial workers, they just use simple lenses. One of the first people to observe objects below 0.1 millimeters and describe what he saw was Robert Hooke. And he looked at cork from wine corks. And he saw that the cork was made up of little boxes. And he is the first person to have referred to those boxes as being cells. What he was looking at, of course, were the remains of cells, the remains of living bark, which had died and then formed cork. But he was the first one to use the word cell. And in fact, the word cell is tremendously important in biology because it defines life. All living things are defined by the fact that they are cellular. And from this arose later on, I'll just describe another figure a little bit later on, who showed that all cells are derived from other cells. All cells arise from another cell. So the initial observation of the cell, well, they didn't really put it together with the structure of living things. Hooke thought that living things must all be made up of cells, but he didn't really develop the theory much further. It remained for a Dutch merchant called Anton van Leeuwenhoek to provide the first descriptions of living organisms which were below 0.1 millimeters and were invisible to the naked eye. These things, these very small things, had probably been observed by other people, but van Leeuwenhoek has the distinction that he actually documented what he saw. He drew, as you can see here. My mouse is working. You can see here, he drew very carefully what he saw. And these are the very first drawings of bacteria. And he was extremely skilled at what he did. He observed all of the different shapes of bacteria that exist: rods, cocci, spirilla, and spirochetes. We'll hear all about that next week, all those different kinds. But he is important because he documented what he saw, he wrote down, and he corresponded his findings to other scientists. Now, he used the very first true microscope. It was not a compound microscope. It didn't use combinations of lenses. It used a single lens, which he ground, but he was very, very skilled at grinding lenses. This was an almost spherical lens. It could magnify very high. It had a high magnification. But the problem with that is how do you use it? You hold it in your fingers, and then what do? How do you have to try and, the problem is you have to bring the object really close to the lens in order to visualize it. And you have to have a way of focusing. It's very difficult to hold a tiny lens like that in your fingers and hold an object somehow and bring it and look. He developed a little mechanism for doing so. And here it is here. This is the first true microscope. It consisted of a plate which held the lens there. It had a pin, and on top of that pin, he could mount a drop of water, a drop of water. Van Leeuwenhoek looked at everything, I must warn you. He looked at everything. He looked at water, seawater, blood. He looked at semen. He looked at his own diarrhea. He looked at all sorts of things and was the first person to describe what he saw in those fluids, sometimes to the horror of observers. But nonetheless, he would mount a small drop of whatever he was looking at on the top of this pin. and he could move that pin up and down so that it was perfectly centered in the lens, and he could move the pin back and forth as well to bring it into focus. First, this is a microscope, a true microscope. It had to be used in bright light, and here he is using it, looking into the sun. But with it, he'd be observing from that side, of course. And he was the first person to describe bacteria. He was also the first person to describe human sperm. He was the first person to describe many pathogenic organisms, such as those which cause diarrhea, because he looked at diarrhea and he saw the organisms. He was a hugely prolific and very, very robust observer, and really the first true microscopist. And he's regarded as being the father of microscopy. Other people were amazed at what he'd seen, and they used his method and very soon developed other forms of microscopy, other forms of microscopes. And the principal development was the compound microscope, which we still use today and which you will, of course, be using in lab. So Van Leeuwenhoek called these little things that he saw them not only as static objects, but he saw them swimming around. He observed them clustered together. He saw them feeding, doing all sorts of things, and he called them animalcules and believed that they had their own natural history within the fluids that he observed. Other people differed from that view, and they believed that, in fact, fluids and various other things could spontaneously give rise to living organisms, and that life is generated continuously. So, for example, even somebody as thoughtful and skilled as Aristotle believed the swallows were born from mud. Because if you watch a mud flat, you will see swallows swooping down to gather mud to go off and make their nest. And sometimes it looks like they erupt out of the mud and fly off. That was a common belief. So here is a very simple one. These are people who believed in spontaneous, this is called this theory of spontaneous generation, that life arises continuously from inanimate objects. And the people who believed that were called the vitalists. And they believed that there was, in fact, a vital force which saturated all of creation. So even if something was inanimate, mud, meat, whatever, was inanimate, it could give rise to life because it was saturated with the vital force, and the vital force drove matter to produce life the whole time. Now, the danger of this is that it's completely unscientific. It's an hypothesis, but it's a hypothesis which can be used to explain all sorts of observations that have nothing to do with spontaneous generation. And it sat like a dead weight on science all the way through to the middle of the 19th century. It sat on a dead weight on science because it stifled scientific debate. Every time a valid scientific hypothesis was raised and explained, and experiments started to be done, then the vitalists would come along and say, oh no, it's just because of spontaneous generation. You're just observing spontaneous generation. You're just observing the vital force in action. So the people who opposed that idea were the biogenesis believers. And they believed, the biogenesis people believed, that living cells and organisms only arise from pre-existing cells and organisms. Cells arise from other living cells. And even Van Leeuwenhoek disliked spontaneous generation, and he attacked one of the simple things that spontaneous generationists always said, that rotten meat, for example, gave rise to maggots because of spontaneous generation. And if you observed that there were eggs on meat, as Van Leeuwenhoek did, he saw flies lay eggs. The spontaneous generation people said, no, those eggs actually arose from the meat in contact with the flies. There's an example of how spontaneous generation can be used to explain even simple observation. So it took some considerable experimentation, starting with simple experiments like this one. This is an experiment by a man called Francesco Reddi, an Italian. And he attacked this idea of rotten meat giving rise to maggots very simply. He took jars, he put meat in to rot, and he covered one of the jars with gauze and observed what happened. And of course, we instinctively know what happened. In the open jar, maggots developed. In the closed, covered jar, no maggots developed. The meat went rotten in both because, of course, we know that bacteria get through the gauze and are able to infect the meat, or in fact, the meat was probably infected with bacteria right at the very beginning when they put it in the jar. But the spontaneous generation people were not satisfied with this. Even though it looked to many observers like this proved that spontaneous generation did not occur, they said, no, all you've done is you've interfered with the vital force. You put gauze over the top of the jar, you interfered with the vital force. The vital force wasn't able to reach its full potential because you covered it, you impeded its activity. And so the meat went rotten, but it didn't develop maggots. They can never be satisfied until this very brilliant scientist that we're going to hear a lot about, called Pasteur, Louis Pasteur. And Louis Pasteur was the first scientist to definitively prove, and it took all the way until 1861 for him to do so. And Louis Pasteur conclusively disproved the idea of spontaneous generation in a very elegant experiment. And it was this. So Louis Pasteur had worked extensively on the processes of fermentation. And he knew and understood that, in fact, there were different organisms which could cause fermentation. He had initially worked on wine. That was how he got his start. He'd studied science at the Sorbonne, but he was conscripted by the winemakers of France to examine the processes of fermentation, which they did not understand at all. They just used natural fermentation proceeding. They would harvest grapes, crush them, put them in a vat, and leave them to ferment on their own. Now that has worked very, very well for many, many thousands of years. That is how we make alcoholic brews of one sort or another. But it is unpredictable. Sometimes things went wrong. For example, sometimes a batch would ferment and go straight to vinegar. It would be spoiled. Sometimes it would develop foul tastes. Sometimes it just wouldn't ferment properly. It would ferment too slowly, or it would ferment too quickly. There was a certain degree of unpredictability, even though they used the same methods, crushing and over and over and over again every year. It was unpredictable. They conscripted Louis Pasteur, who already had an interest in microbiology, in these small things that couldn't be seen with the naked eye. And they conscripted him to work on the wine, and he discovered, first of all, that the principal agent of fermentation of wine is, of course, a yeast. Yeast is a microbe, but it is not a bacterium. But he was able to purify yeasts, and not only purify, but actually kind of pedigree them. He could produce yeast strains which did different things. And he observed that different wines, different vineyards, in fact, had different yeasts that were responsible. And he began to refine the process of fermentation. He began to advise winemakers of how, first of all, to encourage the growth of yeasts which were beneficial, and to discourage the growth of bacteria which were detrimental. It was very often bacteria would cause problems in the wine, impede the fermentation, or add foul taste to the wine. He was able to advise winemakers how to refine their processes and, in fact, how to pedigree their yeasts so that they knew much better what they were dealing with. So he's very famous and very, very popular person in France already. He established a laboratory where he did an enormous amount of work. We'll hear more about it in a minute. But here is how he disproved the theory of spontaneous generation, knowing that liquids containing nutrients are subject to fermentation, to the development of microorganisms. He theorized, first of all, microorganisms are everywhere. They are in the air, they're in the water, everything else all around us. He thought it was possible to sterilize liquids by boiling. But he was, for the most part, he was correct. You can very often sterilize by boiling, but it doesn't always work because bacterial spores are resistant to boiling. In this experiment that we're going to talk about, that is irrelevant. He didn't have to deal with, he wasn't dealing with bacteria that produce spores. So here is what he did. He developed a special set of flasks. These are flasks in which he put a broth. A broth is simply an aqueous solution of nutrients. And he did beef broth. He put in these flasks. The flasks have these long necks. And he would put the broth in and then boil it. Now, some of the flasks he left like this with their long neck, standing straight up, boiled. Other of the flasks that he boiled, but he had bent the necks into these shapes, into this, it's called a swan neck. And these are called the swan neck flasks. And when he boils the fluid here, what is going to happen is the steam is going to pump out of this here. It sterilizes the fluid and it sterilizes the inside here. Now, he reasoned that the air has microorganisms in. Those microorganisms, they don't fly around. They don't have wings and jet propulsion or anything. They are at the mercy of drafts of the air itself. Very often they are carried on flakes of dust. In our case, very often on skin flakes that drift around in the air, but they have weight. And in still air, they will settle. And if you have a flask of broth, for example, and it has an open neck like this, microorganisms can settle from the air and settle in the broth and infect it, even if you sterilized it by boiling. Eventually, if it's exposed to the air like this, where dust particles can settle on the, it will become infected and it will eventually start fermenting and rot. In this flask, he had sterilized the fluid, sterilized the neck, but it had this S shape. What is going to happen? This is open to the air, but particles settling here are going to be trapped here. They can't get around here. There's no air draft. There's no draft to move the particle. It can't go on its own. It can't fly. This is not wet. It dries out. So any particles settling here, this is his reasoning, any particles settling here are not going to be able to make their way there into the broth. And if they can't make their way into the broth, then they can't ferment it. And that's exactly what happened. In flasks with straight necks like this, or some of them, he broke the neck of the flask, microorganisms very rapidly settled and fermented the fluid. In these swan-necked flasks like this, microorganisms did not settle. The broth remained sterile and clear, and it did so not for weeks or months. It did so for years, decades, and then hundreds of years. And if you go and visit the Pasteur Institute in Paris, you can still see these flasks still sitting there, completely sterile, the flasks that Pasteur set up. Now, what did the spontaneous generation people have to say? There is no impediment to the vital force, right? To stop the vital force acting on this fluid. None whatsoever. It's open to the air, so there's plenty of air, plenty of ways for vital force to make its way in and out if there was a vital force. But there is no vital force. There's no vital force to do anything. Spontaneous generation is a complete myth. And instead, what we require for fermentation, or what we call infection, to occur, we have to have microorganisms. And the microorganisms have to make their way from somewhere, from a source of infection, to the site of infection. And that is an important principle, not only for whatever we're doing, cooking, maintaining foods, whatever, it's also an important principle in health. We have to have a source of infection. These infectious diseases do not arise spontaneously. This is becoming incredibly important in this day and age, believe it or not, because there are those in very prominent places, in positions of power, who manage health systems, who do not believe fully in the cell theory that cells all arise from another cell. We have to deal with this in the current day and age. Okay, so Pasteur disproves the spontaneous generation theory. If we are looking at microorganisms, no matter what circumstances, those microorganisms have arisen from somewhere else. They have arisen from other cells. And if we want to understand their origin and their natural history, we have to understand where they come from and how they got where they are. So the conclusions based on everything that Van Leeuwenhoek and everybody else had so far observed all the way up to Pasteur was that microorganisms are present, first of all, as living organisms. They are living things. Second of all, they're too small for us to see, but that doesn't mean to say they don't exist. They are everywhere. We say they are ubiquitous. We have microbes everywhere in air, water, soil. In fact, they have even recovered microorganisms from rocks miles deep in the Earth's crust, believe it or not. So they are literally everywhere. And this is the theory of biogenesis, that every cell arises from a pre-existing cell. And the first person to formulate this idea was in 1858. And this is a famous scientist called Virchow. And we recognize from his work, from the work of all these other scientists, Pasteur, everybody else, that the cell is the basis of life. All life on our planet is based in that single unit of the cell. And every single cell arises from another cell. The only exception to that we don't fully understand is the development of the very first functional cells. But from those first functional cells, once those first cells had arisen, all other cells arose from them. That is tremendously important because the very first cells that evolved were simple cells. We'll talk more about their structure next week. But they were bacterial. The very first cells which arose were bacterial cells. And all other cells have arisen from them through the process of evolution to produce the huge variety of cells and organisms that we see around us today and that have existed through all of the time that life has existed on Earth. Okay, so these scientists, especially Pasteur, developed the first true scientific basis, free of the strictures of spontaneous generation and ideas like that. And really, Pasteur is regarded as being the father of microbiology. Quite apart from his disproving spontaneous generation, he did a huge amount of work in many fields of microbiology. And he was the first, as I've said, to show that microbes were responsible for fermentation. Second of all, he worked in his work on wine. He discovered a very important principle, one of the most important principles in public health, in fact, at that time, which was that cells could be killed by heat, and that if you did it carefully, you could kill off pathogenic organisms preferentially. Now, the reason for that is that pathogenic organisms, human pathogenic, mammalian pathogenic organisms, tend to have a rather narrow temperature range. They are mostly adapted to 37 degrees Celsius, which is the mammalian, overall, the mammalian body temperature. And pathogenic organisms are largely adapted to that. They operate best at 37 degrees. And if you raise the temperature above that, then you can kill them. So this is a principle that Pasteur discovered, that if you took fluids and you warmed them up and you kept them above, I think it's 65 degrees Celsius for about 20 minutes or so, it would kill off pathogenic organisms. It would largely kill off most of the bacteria, not all of them. But this was important because the same principle came to be applied, especially to milk. In the 19th century, in preceding centuries, milk was a very important food source for human beings, but it was also the agent of carrying disease. There were various diseases that we'll hear about when we talk about bacterial disease, but amongst them, tuberculosis, bovine tuberculosis, same tuberculosis that cows get, but we can suffer from as well. Other diseases like brucellosis, these are all bacterial diseases which milk can transmit. And Pasteur discovered that if you pasteurized, if you heated milk up gently, kept it at that temperature for a certain length of time, and then lowered the temperature, you didn't affect many of the properties of milk. It still tasted like milk. It still behaved like milk. You can make cheese and all sorts of things with it. But it was no longer an agent of disease. So, and he was responsible just from that simple observation. He was responsible for saving many, 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 many thousands upon thousands of lives down the centuries. So here's the process itself. And you can see it eliminates a whole variety of different organisms. So now, here is your first little bit of homework, okay? And that is, I want you can use AI or whatever you like, but go please and look up this name, Ignaz Semmelweis, and find this graph or use the one that I've given you here. Because for your first piece of homework, I want you, please, to tell me the story of Ignaz Semmelweis. And especially, I want you to explain this graph here, and I'll point out to you that here's the very important, this line here is the very important line. This is where Ignaz Semmelweis, I will tell you this simple fact about him. This is where he got the hospital that he worked in to start washing their hands. Now, Ignaz Semmelweis was a tremendously important historical figure because he was the first person to suggest that in hospitals and healthcare settings, the principal agents of transmission of disease were the hospital workers themselves, acting on the knowledge that pathogenic organisms arise from other pathogenic organisms, that infections are caused by moving organisms from a source of infection to a site of infection. He attempted to intervene in that transmission. Tell me the story. It must, you can use whatever source you like, but it must be handwritten. I do not want typed submissions. It has to be handed to me on a piece of paper written in pen. Okay, that's a piece of homework. I haven't quite decided when, but I'll tell you in class. So one of the things about Semmelweis's work was people did get to hear about it. It's a sad story, but people did get to hear about it. And it confirmed for many people who already knew something about or were paying attention to what Pasteur was saying about microorganisms being everywhere, and that a infectious disease arose because a source of infection was transmitting microorganisms to a site of infection. This all sparked the idea in people that disease itself was caused by microorganisms. Now that to us seems so obvious, but it was not at all obvious to people in those days. It had to be actually proved that microorganisms do in fact cause disease. And this man here is equally important to Louis Pasteur. And Louis Pasteur, this man called Robert Koch, he was a German. He was an intense, fierce rival of Pasteur's. They vied with one another to be the most famous microbiologist. And that competition, they disliked one another very much. But that competition was very healthy because it drove scientific development. So Robert Koch was a bacteriologist. He looked specifically at bacteria and those bacteria which cause disease. And he had this theory that in fact any disease is caused by a specific bacterium. Seems obvious to us, but it was certainly not obvious in those days where people doubted the ability of bacteria to cause disease at all. And Robert Koch lit on this one particular organism, which is called Bacillus anthracus. You will hear more about it later. But remember that name for the moment. Bacillus anthracus is the name. And he believed that this bacterium caused the disease of anthrax because he was able to extract that bacterium from animals which died of anthrax. Anthrax is a very terrible disease, and it caused massive losses of sheep in particular, but also cattle. So agriculturally, it was very important, but it is also a very dangerous disease in human beings, and very commonly where a flock of sheep or whatever was infected with anthrax, farm workers would be infected with anthrax, and in one particular form of anthrax is invariably fatal. So it's a very, very serious issue. And Robert Koch believed that it was caused by this bacillus, which he consistently saw. And he developed an experimental method for proving that Bacillus anthracus caused anthrax. And in that doing that experiment, he erected what are called Koch's postulates, the steps that need to be gone through to demonstrate that a specific microbe causes a specific disease. And he also, in doing so, began to show ways in which the transmission of these could be avoided. And the experiment ran like this. First of all, he observed all of the instances of anthrax, where sheep died, etc. He realized that they all showed the same symptoms. You could recognize the disease of anthrax by its symptoms. And when he did postmortems on sheep which had died of those, showing those symptoms, he consistently extracted this organism here. And this is a bacillus. Here is the cell here. The dark here is the cell. And very characteristically, they commonly have a spore. This clear area here is a spore, a bacterial spore in the middle of the cell. And so they're very easily recognized. So he would extract body fluids, examine them, and extract these bacteria. He found that he could cultivate these bacteria. They grew in culture. If you provided them with nutrients and the correct temperature, they would grow and multiply even in the absence of the host. And so that's what he did. He established pure cultures. He established cultures where only this bacterium grew, none other. And he would then inject that broth into an experimental animal. And when he did so, the animal would get ill and would die. And he then extracted and purified the identical microorganism from this dead animal. So there are the steps: finding the source of the infection, isolating the agent, passing it through an experimental healthy subject, and producing the same disease. And when the disease was established or concluded with the death, then extracting exactly the same organism again from them. These are the steps of Koch's postulates, and they are still used today in showing that a particular microorganism produces a particular disease. These are the steps that you need to go through. It is not always possible to do so, and I'll give you a clear example of that. Sometimes it is not ethical. For example, in diseases which only infect human beings, you cannot extract the organism, microorganism, or whatever, the infectious agent. You can't extract it and then inject it into a healthy subject to see if it causes disease, and you can re-extract it to fulfill these steps here. For example, the HIV, human immunodeficiency virus, which causes AIDS, that is a purely human disease. You can't go around extracting from people with HIV and injecting it into people without. But unfortunately, in the case of HIV, that in a way has happened because a common route of transmission has been injectable drugs. So inadvertently, the Koch's postulates for HIV have been proved, but not deliberately. There are many other instances where it is simply not ethical to proceed through all of Koch's postulates. But with sufficient association between bacteria, between the causative agent, you can do it for viruses as well as bacteria, but with sufficient association between an organism and disease, and with a microorganism consistently producing the same symptoms in a subject, we can assume the infectious association. But we always strive to satisfy all of Koch's postulates to prove that a specific microorganism causes a specific disease. So all of these ideas, bacteria are ubiquitous, microorganisms are ubiquitous. Microorganism, you need a source of the microorganism in order to produce an effect. All of these things began to build to this idea of that in many diseases were actually infectious by nature, and that if you could interrupt that chain, source of disease of organism to site of infection, if you could interrupt that somehow, you could prevent disease from developing. So here is a very important figure. This is one of the most important historical figures in human health, in actual fact. And this is Joseph Lister. Lister paid attention not only to what Koch, Pasteur were doing, he had also heard about Semmelweis. And he developed a system within his hospital. He was a surgeon, and he began to sterilize the surgical environment that he was operating in. Now, in hospitals at that time, this was the problem that Semmelweis encountered. Surgeons and nursing staff did not wash their hands. In fact, surgeons took great pride in the fact that the dirtier their surgical aprons were, the more experience they had. So it was a badge of honor to be dirty. The same thing went for their surgical instruments. They did not boil or sterilize surgical instruments. They just moved from one patient to another with their surgical instruments. And going to hospital was, in fact, the most dangerous thing you could do if you were ill. And surgery was an absolutely brutal undertaking. And surgeons routinely had a team of helpers who were huge burly men who held down a patient because, of course, there was no anesthetics. There was no sterilization of a surgical site. The surgeon operated as swiftly as possible, and that usually entailed brutally cutting or sawing or doing whatever, and then trying to staunch the bleeding, often with dirty bandages, dirty rags, whatever, and then move on to the next patient. And it is no surprise that one of the most dangerous things you could do if you were ill was to go to a hospital, because very few people survived. And for example, the mortality rate of women giving birth in hospitals was much, much higher inside a hospital than it was if they gave birth at home. But Lister changed all that, because he began to sterilize his operating room, and at much the same time, the first anesthetics, the use of ether in particular, and then of chloroform, began to be used in operating rooms so that they could work slowly. And Lister, what he did was he sprayed everywhere. He had inside his operating rooms, he had an apparatus that produced a mist of carbolic acid. And carbolic acid is a very nasty substance, but it does sterilize. It does kill off most microorganisms. So he had a relatively sterile operating area, and he could, once anesthetics developed, surgeons began to be able to work slowly and carefully rather than brutally. And the mortality rate within hospitals began to plummet. And for the first time, you didn't take a huge risk just by going under the knife, under really, really unsanitary conditions. So his demonstration was exactly what Pasteur had said. The microorganisms are in the environment. In a hospital environment, organisms which cause disease tend to proliferate because that's where sick people go. And the processes of treating them, if you are not observing sterile technique, the processes of treating them tend to spread them. So Lister and Pasteur, these people all demonstrated that germs come from the environment, and if you control this movement from a site of infection, from a source of infection to the site of infection, through interventions like cleanliness, hand washing, all of these sorts of things, then you can break that cycle. You can actually improve the chances that you will not acquire microorganisms. So I should also mention that one of the really important things that Koch did was to develop systems to actually culture bacteria in pure culture. Now, this is what this means. Pure culture is a system whereby you can be sure that what you are dealing with is one particular microorganism alone. Now, that sounds like something relatively simple. It's not. It's not simple at all. Up until a certain point, microbiologists depended upon broth culture for cultivating microorganisms. An aqueous solution of nutrients. But the problem with broth culture is a broth culture with a pure culture in it may look exactly like a broth culture that has a mixture of bacteria in it. It's very difficult to tell which is pure and which is contaminated. And if you're only dealing with broth culture, it's also very, very difficult to purify. Let's say you were cultivating a particular organism, a really special organism in broth culture, and you cultivate, and one day that culture got contaminated, and you didn't realize, you carried on until you had a big mixture of other organisms together with the one you were interested in. How would you then extract the organism that you are interested in? It's very, very difficult if you're only using broth. But Koch developed a system whereby he created solid media. So this here is a plate, and in this case, it is an agar plate. It's a plate of nutrient broth solidified with agar. And these are the plates which you're going to be using in lab. Once you have a solid plate like this, it's very easy to purify organisms on it, because the bacteria tend to produce colonies which have distinctive physical characteristics. You can recognize a particular organism by its colony, by its color, by its shape, by the way it grows, and other characteristics. So this is all that you do. I'll just do some. You're going to do this in lab, but I'll just tell you simply. Here is let's say you have a mixed culture. You take a little bit of that mixed culture, you call it an inoculum. You spread your inoculum on the solid medium here. You just smoosh it all out. Then you take a loop, a microbiological loop, and you take a swipe across that. What are you doing? You're just taking a little small sample of that, and you're spreading it out. As you drag your loop along, fewer and fewer and fewer of the microorganisms most of them are deposited behind. Fewer and fewer and fewer of them. Then you take another swipe, and you begin to smoosh out again. Every time you do this, you're taking less and less and less and less, until in the end, you spread so few bacteria that when you spread them out, the cells separate one from the other. This colony has arisen from a single cell. This colony here has arisen from a single cell. if they, in this case, they look like they're sharing characteristics. Once you do this, you can easily recognize your microorganism. And if you want to purify a culture from something that's mixed, you just pick out one of the colonies that has the correct characteristics. And then you can take it onto another plate and you can amplify it. You can cultivate it on that plate. So this is a very simple way to purify cultures and to maintain pure cultures. And this is something you are going to learn. And this is all the responsibility of Robert Koch. I should also mention, by the way, that the method using agar was developed by the wife of one of Koch's workers. She was an American and living in Germany with her husband. And she used to use agar to make jellies and jams in her kitchen. And she suggested to her husband and then to Koch that they use agar in order to make solid meat. Her name was Fanny Hess. And remember, when you use an agar plate, it's thanks to Fanny Hess that we do so. Okay, so here are the huge developments in the relationship of human beings to disease. First of all, we've discovered that many diseases are caused by microorganisms, that the transmission of microorganisms from a source of infection to the site of infection can be interrupted. That in itself, huge implications for public health of human beings. But here's the other major impact on human health, and that is the science of vaccination. So vaccination is a process whereby we teach our immune system to recognize characteristics of a microorganism before we are infected with it. So that when we really do get infected, our immune systems immediately recognize the infection and wipe it out. So this, the process of vaccination has a very long history. Its roots lie in two sources, first in ancient China, but also in ancient Arabian countries. It was Arab scientists, Arab doctors, first developed systems whereby they taught immune systems to recognize disease before it arose. So the disease that everybody was fascinated and terrified by was smallpox. The Arab doctors actually, what they would do is they would go to people who were recovering from smallpox, and they would take scrapings from healing sores, and they would use those to inoculate people and cause a mild case of smallpox. Well, they hoped it was mild. And in about 60% of cases, it was. And once people had recovered from their mild smallpox, they would be immune for life. The problem was 40% suffered serious smallpox and died or were hideously disfigured for the rest of their lives. This is what smallpox looks like. It's a terrible, terrible disease. It was one of the plague diseases that caused millions of deaths worldwide on a regular basis. So people were terrified of it. But this gentleman, Edward Jenner, heard of something in England that it piqued his interest, which was that milkmaids did not suffer from smallpox. Milkmaids never suffered from smallpox, even during epidemics. And the reason for that, what he discovered, was that most milkmaids in England, in the course of milking cows, would acquire a disease called cowpox. And cowpox causes a very mild response in human beings. Usually it was simply a little rash of blisters on the fingers that they used for milking. And they would recover from that very easily. And once that had happened, they were immune. He made that association. this was legend that people spread about milkmaids. He went and investigated it and discovered that this was a regular occurrence. Milkmaids, that cows got their own form of pox. Milkmaids picked it up, and he started to collect serum from those cowpox blisters. The blister itself is called a pox. So he would go and collect serum from those blisters of cowpox, and he would use that to inoculate people. He would scrape it into their skins. And they got no more than a small sore where he did the scrape, and they were then immune. That protection is immunity. What he has done is he has, instead of using smallpox itself, the way Arab and Chinese doctors did, instead of using smallpox itself, he would use an entirely different organism. But it's sufficiently similar to smallpox that our immune systems confuse them. So if you develop immunity to cowpox, if you're ever exposed to smallpox, your immune system thinks it's the same thing and mounts a huge response and prevents an infection. So that is the principle of vaccination. And the word vaccination comes from its source, vaca, which means cow. And it's because Lister used cowpox as his source of organism. Now, that principle of vaccination was established in the 1600s. I'm sorry, in the 1700s. And workers like Pasteur knew about this very well. And Pasteur went on to develop vaccination for all sorts of diseases, most importantly, rabies. That was one, but various others. Anthrax was another. They developed vaccinations for these different diseases using the same principle. And what they would do in many cases would be they would take a disease-causing organism and they would weaken it. They would pass it through a whole series of infections in experimental animals. In Pasteur's case, he used rabbits. And as it passed through, each time it was infected a new subject, the organism would be weakened. And in the end, they would end up with a very much weakened infectious agent, which they could use to vaccinate. So nowadays, we have all sorts of vaccination strategies, but this, and we'll talk about that a little bit later on in the course. We'll talk about mRNA vaccination, for example, that was used for COVID and is now being used for influenza and various other things. If we can get around what is being done by Health and Human Services. But the principle is always the same. What we do in these systems of vaccination is we take something, we take part of an organism, we take a weakened organism, whatever, and we teach our immune system to recognize disease before we actually are infected with it. So that when we really are infected, our immune system immediately responds. So all of those, if you want to think about it, all those things, hand washing, sterile operating rooms, vaccination, these are all preventative, very good preventative mechanisms. But what if you actually acquire a disease through a microorganism? What can you do then? So this was a huge area of inquiry, and of course has been for all the time that human beings have existed. We have sought cures for disease, but for most of human history, the cure has usually been worse than the disease. And for example, for much of the 15th, 16th, 17th century, for example, very, very toxic compounds were used to try to treat disease. So, for example, mercury, arsenic, strychnine, all of these terrible poisons were used to try and cure disease, and very often ended up killing the person who's suffering from the disease. The cure was very, very often worse than the disease itself. And there was always this dream that there would be some way of directing an agent, a chemical agent or whatever, against a particular disease, against a particular disease-causing organism. But it was really only towards the end of the 19th century, the beginning of the 20th century, that this really did happen. And the treatment of disease with chemicals is what's called chemotherapy. And the development of effective chemotherapies was very, very recent. Actually, in our terms, our history, very, very recently, very recent. And it's the discovery of the first effective agents ushered in what's called the second golden age of microbiology. The first thing to note is that there are various ways that this can be done. The first is by synthetic drugs that can be produced. This other way is through antibiotics, and antibiotics are compounds which are derived from living organisms, from bacteria and fungi principally, which kill off other microorganisms. And the first synthetic drugs actually are those which were extracted from biological systems and then modified. For example, the use of the drug quinine is very old, very ancient. But that quinine, in fact, was one of the very few effective treatments, for example, for treatment and prevention of malaria. There was almost no other effective drug in human usage, almost none. Until this scientist, Paul Ehrlich always speculated about a magic bullet, a single drug which would kill only the microorganism causing disease and would not affect the human being who was taking it. That was his ideal magic bullet. Magic bullets seldom occur. We usually have, associated with any chemotherapeutic agent, we usually have some degree of risk. But they can be refined so the risk is less than the risk of disease, and then it's worth taking. And in 1910, Ehrlich developed the first effective synthetic drug. It actually was based on arsenic, but it was relatively non-toxic. It was called salvarsan, and it was used to treat syphilis. They did try using it for treating all sorts of other things, but its principal development was to treat syphilis, and was relatively successful. In the 1930s, the first synthetic sulfonamides were synthesized. These were regarded as being miracle drugs. They were antibiotic in the sense that they killed off living organisms, that they were specific. They were not antibiotics derived from living organisms, as the true antibiotics are, but they were highly effective against many, many different organisms, such as tuberculosis. The problem with them was that they had many side effects, and that organisms very, very rapidly developed resistance to them, and their effectiveness soon waned. Within a decade, 2 decades of their development, they were already pretty much useless for treating many strains of tuberculosis and other diseases. So they had some effect, but that was extremely limited. But in 1928, a scientist called Alexander Fleming discovered the first true antibiotic, and this was penicillin. I'll just show you here. This is exactly how he discovered it. This is the plate which actually, the famous plate where it was first demonstrated. He was going on holiday. He was working on an organism called Staphylococcus aureus, which is a major cause of infections in hospitals. He was working on Staphylococcus aureus, and here it something's happened here. Can't find my mouse. Anyway, have a look down there at the bottom, and you'll see it says penicillin colony. And around the penicillin colony, you'll see a clearing. It's not a complete clearing, but you'll see that there are just small colonies of Staphylococcus aureus. But as you move away from the penicillin colony, the Staphylococcus aureus colonies get bigger and bigger and bigger. And this fascinated Fleming. He did write it up. He did nothing to develop the idea further, but he speculated that in fact there was that what had happened. Oh yes, I'll go back. Okay, here's the penicillin colony. Here's a zone here of relative clearing where the Staphylococcus aureus is inhibited, and then as you get further away, they get bigger and bigger and bigger. He speculated penicillin is releasing a compound to inhibit the growth of bacteria. And the colony itself is from a known fungus called Penicillium, and he speculated this compound, he called it penicillin. This is a natural phenomenon. Fungi and bacteria as well will release these compounds to inhibit the growth of other organisms in their environment, which might compete with them for nutrients. So it's a natural phenomenon. Many, many, many bacteria, many, many fungi and other organisms will produce these kinds of inhibitory compounds to try and stifle the competition of other microorganisms. Fleming did nothing with it. He didn't proceed to say, okay, how can this be used in treating disease or anything else? He didn't make that kind of association. But during the Second World War, the need for antibiotics was enormous for treating battle wounds and this sort of thing. And workers who knew about Fleming's work actually developed ways of culturing penicillin fungus in bulk, in broths from which they extracted penicillin and used it as an antibiotic. It was spectacularly successful for quite a long time, until it was discovered that penicillin also produces resistance in microorganisms. But by that time, they had started to discover many other compounds produced by microorganisms as antibiotics. And they also began to synthesize other forms of penicillin. So for a very, very long time, antibiotics were the war chest of public health and of treatment of disease. But increasingly, we're finding problems with the development of resistance to these antibiotics. So now the fields of research moved from antibacterials to looking for antiviral compounds, which are very rare. There are still very few compounds which combat viral infections. And in the 1950s and 1960s, the next major development in microbiology was the discovery of DNA and the ushering in of the age of molecular biology, which many people call the fourth golden age of microbiology. So when we go through our course, we're going to encounter 4 ologies. The first, our major area, is going to be bacteriology, the study of bacteria. Very minor, we will only have a brief reference to it. Mycology, the study of fungi. Although fungi do cause human disease, we are not going to discuss it in great detail. Nor are we going to discuss parasitology, which is the study of eukaryotic microbes. And this includes some macroscopic organisms because their larvae and early stages are microscopic. Protozoa, parasitic worms, all of these sorts of things are parasites. And the fourth major area that we will be discussing in this semester is virology. And virology is the study of viruses. Viruses were discovered very late. They were discovered because scientists realized that even when they sterilized broths and things like that, by passing them through very, very fine filters, which would filter out all the bacteria, there were still infectious agents present in the broth. And this was in particular the case in dealing with plant diseases. And the very first virus to be identified was a virus which caused disease in tobacco. Tobacco was commonly used as an experimental plant in scientific laboratories. And they thought all the diseases that occurred in bacteria were caused by bacteria, and then discovered that even when they screened out all of the bacteria, there were still infectious agents. And this was the first observation of viral infection. Okay, so that is my brief and hurried introduction to the history and study of microbiology. And I will see you on Monday.